So I'm Paul Nioi. I'm Vice President of Discovery Research at Alnylam Pharmaceuticals. I'm going to do three things today. The first is I want to give you a little bit of an insight into Alnylam and particularly the mechanism through which our drugs work, which is quite different from traditional small molecules and monoclonal antibodies. So I'll spend a couple of slides describing that. But the meat of the presentation is really on two programs that are, one of which is in the clinic, the other one is preclinical, and they're both designed to treat NASH. So I'll give you some insights, and I believe the first program I'm going to talk about, we've actually been working with Richmond Pharmacology on one of the parts of that trial, so you may be familiar with some of what I'm going to describe. But let me begin by describing how our drugs work. So Alnylam is a company that was founded on the basis and with the idea of taking a discovery, and that discovery was RNA interference, and turning that from an observation and a tool that could be used in the lab into a drug. And that's really why Alnylam was established and what we've been doing for the last 20 years. It's actually our 20th anniversary this year. And so the idea is actually to exploit a natural cellular mechanism that exists in all cells in your body. And it's designed to sense the presence of double-stranded RNA, which of course many viruses harbor in their genomes. What we do is we hijack that system by introducing synthetic double-stranded RNA into cells in the body. It is then recognized by a complex called RISC, or RNA-induced silencing complex. And that enzyme unwinds the double-stranded RNA. One of the strands comes out, and the other strand goes into RISC, and RISC then proceeds scanning all the RNAs in your cell, looking for matches. And when it finds a complementary match, it chops the RNA up, it degrades the RNA. So obviously, if you have a viral infection, it's a very powerful way of clearing the virus from your system. But if you can imagine, you could program these short double-stranded RNAs and give them the sequence of an endogenous gene, you can actually direct this machinery to degrade a gene of interest, a target gene. And so that's the way that our drugs work. We act by silencing the expression of disease-causing genes within the cell. You'll also hear the term from silencers is kind of the vernacular that's often used to describe RNAi therapeutics. But this is the mechanism through which they work. And we've spent really the last 20 years trying to crack. Well, our RNA is very inherently unstable. When you inject it into the bloodstream, it's rapidly degraded. There are many, many problems to overcome in order to turn this discovery into a therapeutic, and that's where we've been focused. Interestingly enough, the observation of RNA interference was first made in plants, and this whole journey started in 1990, where Napoli and Jorgensen were studying petunias. And they had this idea, well, could we make a petunia more purple by introducing more of the RNA that encodes the purple gene into the plant? And they tried this, and surprisingly, rather than making the plant more purple, the flowers more purple, it actually made them white. So the color went away, and that was actually the first description of RNA interference. What was happening was they were introducing RNA into the cells of this plant, and it was being recognized by RISC, and it was actually silencing the expression of the gene that gives the plant, the flowers, the purple color. And that was subsequently the inspiration for the work of Andrew Fire and Craig Mello, who initially working in C. elegans and then in mammalian cells, showed that this phenomenon is something that is very evolutionarily conserved. It exists in all cells in mammals. And that was the foundation for Alnylam. It was, okay, we have this very, very powerful tool where we can silence any gene in the genome. There's, this sort of concept of what's druggable really is turned on its head. We can target anything, provided we can deliver the double-stranded RNA to the cells of interest in the body. And that, in a nutshell, has been the challenge for us over the last 20 years. How do you stop RNA from being degraded? How do you deliver it to the right tissues in the body? And the papers that you can see are right along the bottom of the slide here describe in detail, if you want to go and read them, the path that we've been on. It started with an initial demonstration in non-human primates that we could deliver RNAi to the liver of non-human primates and see silencing of genes, to the discovery of lipid nanoparticles, which you're all very familiar with. And they've been a big part of course, 2 of the COVID vaccines that have been approved. But we've used the same technology to deliver double-stranded RNA. And then subsequently, we figured out how to stabilize and prevent the RNA from being degraded, from being recognized by nucleases in the bloodstream, and conjugate it to a simple sugar, which is recognized by the liver, and so it's rapidly taken up by the liver. So we can target hepatocytes very easily now using this system. What I'm not going to talk about today, but is actually really quite pertinent given the last talk and Jörg's opening remarks. In the initial years of Alnylam, once we got into the clinic, we were very much plagued by transaminase elevations in our phase 1 clinical trials. And what turned out to be happening was, that if you imagine a short stretch of double-stranded RNA, it's about 20 to 23 nucleotides in length. Of course, you want it to bind specifically to your gene of interest, but there may be parts, particularly in what's called the seed region, the initial part on the 5' end of the siRNA, that recognize other genes. And the more promiscuous your siRNA is, the bigger the perturbation that you see in the transcriptome, and the more likely you are to see elevations in transaminases. And so we solved that problem by introducing certain modifications to the RNA that destabilize some of these off-target interactions. And we've shown actually with one of our programs, which was to knock down alpha-1 antitrypsin, that in our first clinical study with that molecule, we had transaminase elevations. We went back and modified it to destabilize the seed region. So essentially exactly the same sequence, but with the modification. Took that into the clinic again and saw no transaminase elevations. So now that is kind of the standard approach when we're designing our drugs. So we're very excited to say that we've actually have 3 wholly owned approved products at this time, starting from 2018 when Onpattro or patisiran was approved for the treatment of hATTR amyloidosis. We then had Givlaari approved for the treatment of acute hepatic porphyria. And finally, Oxlumo, which was approved for the treatment of primary hyperoxaluria in young children. We also have a drug which is partnered with Novartis called Leqvio, which targets PCSK9 and reduces PCSK9 protein levels and is used for the treatment of hyperlipidemia, hypercholesterolemia, and was recently approved, I think, last year in the UK. So we've sort of cracked this problem, and now the question is, well, what are all of the diseases we can treat? We can target the liver. Where can we use this technology? And I'm going to focus today, as I mentioned, on non-alcoholic fatty liver disease, but more specifically on NASH. As you heard, there are a large number of patients who have simple steatohepatitis, simple steatosis, but there's a subset of patients that go on to develop inflammation, cirrhosis, and on some occasions hepatocellular carcinoma. You can see the numbers here in the United States. It's actually staggering when you think about this. A quarter of the population in the US have NAFLD. So 96 million people in the US are thought to have NAFLD. Of those people, 5 to 6% have the more insidious form of this disease, where they actually go on and develop inflammation in the liver. So it's around 16 million people. Around 3 million of that population actually have progressed even further and have cirrhosis, and some have further developed HCC. And you can see some of the images at the top here, kind of comparing a liver on the left, where you can see the white kind of globules of fat in the liver and evidence of some inflammatory cells. And then you can see what happens on the right when the liver becomes very scarred over time, and I'm sure not very wobbly by FibroScan in panel B there. There are no approved treatments for NASH, other than we know that losing weight is really good at reducing fat in the liver. So if you stop and you can control your diet, it's a really advisable thing to do, and will certainly go a long way to improving your condition. But there are no effective pharmaceuticals that have been approved for the treatment of NASH. And that's not through want of trying. There are many trials, many failed clinical trials in this area, so it's a very challenging space to be in as well. And it's comorbid with, as I mentioned, obesity, with metabolic syndrome, with type 2 diabetes. So many of the things that we see quite prevalently in the Western world. So, if we're to think about how to treat it, I mentioned in the previous slide, you can kind of think about stages of NASH, or stages along the path to developing NASH. In one case, you simply have the accumulation of lipids, of fat in the liver. So that's one point of intervention. If you can stop the accumulation of fat, or you can reverse the accumulation of lipids in the liver, that's one strategy that could be used. But of course, if you see a patient suffering from NASH, they may be at various stages. So it really warrants intervention at different points in this process. So another approach to think about is, you have this benign accumulation of lipid, but perhaps you can stop that progressing to this low-grade kind of inflammatory condition. And so there's another point of intervention there. And then maybe even if there is inflammation, you can do something to hinder fibrosis. So we've been thinking about this in a way of, what are the targets we can think about in different stages of NASH? And I'm going to talk about two programs today. One is called PNPLA3. I'll describe that in more detail in a moment. The other one, which acts downstream of, it would do nothing to prevent the accumulation of lipid, but it would, we think, be very powerful at preventing the inflammatory component of NASH. And that's a protein called HSD17B13, or 17-beta hydroxysteroid dehydrogenase 13, which is a very interesting protein, as I'll describe, that came from, in fact, both of these came from the study of population genetics. So just a little bit of biology, zooming in a little bit, molecular kind of view of NASH. So obviously you have lipid droplets that form within the hepatocyte, and those are actually quite normal. And there's protein machinery that assembles on these droplets of lipids, and its job is to process the lipid within these droplets. And in fact, both of our targets that we're interested in are present on the surface of lipid droplets within the hepatocyte. So both HSD17B13 and PNPLA3 are found on the surface of these droplets. Interestingly, what has been described is that if you have a mutation in HSD17B13 that causes loss of function, so stops the protein from being produced, it's a naturally occurring mutation, you actually are at lower risk. You have a lower odds of progressing. Should you have fat in your liver, the likelihood of you progressing and actually developing NASH is about half of what you see in the normal population. So that's the first observation, the protective loss of function variant. The case of PNPLA3 is quite different. Here you have a toxic gain of function mutation. So it's a missense mutation, changing a single amino acid in PNPLA3 and putting people at risk of the development of NASH. So there's two different concepts here. In one case with HSD, we want to knock down HSD17B13 and mimic the protective effects of the genetics. In the other case, we want to identify individuals that carry this toxic gain of function mutation in PNPLA3 and treat them by knocking down expression of the toxic protein. So it's two different strategies that we're taking in NASH. I'll begin by talking a little bit more about HSD, and this is the program, I think in one of the parts of our phase 1 study we've done in partnership with Richmond. It's excited to share some of this with you. This is a target that actually came to Alnylam via a collaboration we have with Regeneron Pharmaceuticals. And the original observation came from Regeneron, from the Regeneron Genetics Center, where they were studying a population of individuals in the Geisinger Health System in Pennsylvania. And so they had kind of access to detailed medical records from around 100,000 individuals, patients in the Geisinger Health System. And that included lab test values, it included ICD-10, diagnosis codes. So what they were able to do was ask some questions like, well, and I should also mention they did whole exome sequencing of that whole population. So it enabled them to ask the question, well, is there variation in the sequence of the genome in individuals that are at lesser or greater risk of the development of NASH? And I'm showing you a figure here where they looked at levels of ALT and AST in this population and looked for SNPs, single nucleotide polymorphisms, in the genomes of individuals and whether they associated with altered levels of ALT and AST. And so what you see here is each of the dots on these plots represents a single SNP, a single nucleotide polymorphism in the genome. And then on the y-axis, you can see minus log 10 p-value. So the higher the peak, the more significant the finding is. And this is called a Manhattan plot because the peaks are supposed to look like the skyline of Manhattan. And so they made a really interesting finding, and that was that there was most of these things, in fact, I think all of them, apart from HSD17B13, had been described from similar studies in previous publications. But HSD was new, and they found that there was a significant association with this particular SNP, which was a loss of function mutation, and both ALT and AST. And it was associated actually with lower levels of ALT and AST on average. So they have these measurements across a period of time, however long that person has been part of the Geisinger Health System, and they take the mean of those measurements, and that's what they're assessing here, controlling for things like age, sex, BMI. So that was very interesting. They then went on to show that it is not just a change in ALT and AST that it associated with, it was also lower risk of being diagnosed with a whole spectrum of different liver diseases, including alcoholic liver disease, alcoholic cirrhosis, non-alcoholic liver disease. And you can see on the right of this figure here, the odds ratio per allele. On average, for most of these diseases, it's a 30 to 50% decrease in the risk of developing any of these conditions. And so this is really appealing to Alnylam. We were approached before this publication in the New England Journal to work together with Regeneron on this target, with the idea being, if we are able to deliver an RNAi therapeutic to the hepatocyte, can we knock down the expression of HSD17B13 and in normal individuals mimic these protective effects that we're seeing in carriers of this loss of function variant? That's really the hypothesis that we're trying to test at the moment. So, as I just described, you have the presence of HSD17B13, and I should add, we don't know what this enzyme does. We don't know what the substrate is. We don't know exactly what its role is in driving inflammation in liver. What we do know from various detailed studies that have been done is that it has absolutely no effect on steatosis per se. So there is still accumulation of fat in the liver, but there is less inflammation in individuals that have loss of function. So there seems to be some role for this protein post accumulation of fat, but prior to the development of inflammatory disease in the liver. And so that's our hope, is that we can have a similar type of effect by removing this protein in the liver of patients with NASH. We have developed a double-stranded RNA conjugated to what's called GalNAc, which is a simple sugar recognized by the asialoglycoprotein receptor in the liver. It allows uptake of the siRNA. And this is some data that we generated prior to going into the clinic in non-human primates. You can see two different doses, one at 3 milligrams per kilogram, the other at 10 mg per kg. It's a single dose. And what we did is we ran the study for 14 weeks, and we took biopsies from the monkeys at various time points that you can see in the graph here along the way. And we measured both mRNA, which of course that's what we're targeting. We're trying to degrade the mRNA with our approach, but also looking at HSD protein levels in the livers of these animals. And you can see in both cases, we see a very rapid decline in levels of HSD17B13 mRNA, followed by a drop in levels of the HSD protein. And what's remarkable about an RNAi-based approach is this is a single dose, and the duration of effect that we see is incredibly long. And this is true in humans as well. In fact, our most recently approved drug that I mentioned earlier, Leqvio, that targets PCSK9, is given once every 6 months. And that's because the siRNA gets into the liver and is slowly released from an intracellular storage compartment depot over a period of time. So you get this incredibly long pharmacodynamic effect. And so this really was some of the seminal work that in discovering ALN-HSD that then allowed us to go into phase 1, which is it has just wrapped up actually relatively recently. We're preparing to go into phase 2 at this point. You can see a couple of different parts to our phase 1 clinical study. Of course, our initial study was a single dose, and the goal was really safety and tolerability. What did we see was this, with this particular molecule, did we see any evidence of PK, PD, did we see any untoward effects in any of the volunteers in the trial? And Part A was run in healthy volunteers. You can see the various cohorts that were included. We went all the way from 25 milligram dose all the way up to 800 milligrams, subcutaneous single injection. We have to space the dosing out quite a bit, again, because the drug has a really long duration. So, a single dose, it's not cleared in a few hours, it's cleared in a few months. So, we had to titrate up relatively slowly. And then Part B was a multi-dose study, and that was actually in NASH patients. I'm not going to discuss that data today, but the goal there was really, can we see any evidence of efficacy in these the small cohort, but can we see any evidence of, for example, reduction in ALT, reduction in AST? We were going to take biopsies and see whether we saw any histological improvement in phase 1. I'm very happy to tell you that at least in the Part A study, we saw no adverse events related to the study drug. Any AE that we did see was mild in severity. There were no deaths. We did have one treatment emergent serious adverse event, but that was unrelated to the study drug. It was tonsillitis requiring hospitalization, but that resolved, thankfully. And no clinically significant elevations in serum ALT or other lab or abnormalities, although I don't know what the normal range was. And Jorg, you might know. But there was nothing concerning to report. One thing we do see quite often are injection site reactions, especially at higher doses. You're putting, a lot of drug. 800 milligrams is actually quite a big volume under the skin. So we do, from time to time, see injection site reactions, but these typically resolve very, very quickly, and no ECG findings of any significance. So we're excited about this. Of course, this is just the beginning. As I mentioned earlier, it's a very challenging field to be in. There are many, many examples of failures. There are 0 examples of successes in the development of drugs to treat NASH, and so the proof will be in the pudding once we get into later stage development and see whether we can actually impact the course of the disease. So that's where we are with ALN-HSD. For the final part of the talk, I want to switch gears and talk about another protein, another target called PNPLA3. And this is a really interesting story, actually, that goes all the way back to, I think, 2008, when the involvement of PNPLA3, or patenin-like phosphatase 3, was first described by Helen Hobbs et al. And what we know now is that PNPLA3, and there's a mutation in this gene, and it can cause, this mutation seems to cause the inappropriate accumulation of fat in the liver. And then, of course, you have all the downstream sequelae of fat. So it's predisposing people to the development of NASH. And it's a single amino acid change where an isoleucine in position 148 is changed to methionine. This, again, is a study that was conducted using population genetics, very famous study called the Dallas Heart Study, where they found this really significant association of this mutation with the likelihood of developing NASH. And in this particular Manhattan plot, what they're looking at are liver triglycerides as measured by MRI. On the right, you can see actually that that residue, that isoleucine, is very, very conserved across species. In fact, it only varies in the frog, where there's a valine instead of an isoleucine, but those are very, very similar amino acids. And it seems to be something about that change, going from an isoleucine to methionine, that somehow alters the activity, the function of PNPLA3, and I'll show you a little bit more mechanistic data in a moment, that predisposes to the development of NASH. And interestingly, at the bottom here, if you look at hepatic triglycerides versus BMI versus HOMA-IR versus plasma triglycerides, the only difference that you see is the presence of an increased percentage of triglycerides in the liver. So it's some specific mechanistic difference in hepatocytes that's leading to this accumulation of lipid. There's a number of really interesting studies. It's very, very well studied, very well established as a major risk factor for the development of NAFLD and NASH. What we know now is that there may not be some kind of beautiful kind of mechanistic explanation for what's going on, and it may simply be due to just too much PNPLA3. So what's been described is that this mutation, when you replace the isoleucine with a methionine, the resulting protein is actually resistant to proteolytic degradation in the cell. So it's not degraded as efficiently by the proteasome. The panel at the top shows some data from transgenic mice where they took the wild-type mice and then they introduced the wild-type human PNPLA3 gene into the liver, and then stained the liver with oil red O, which is a stain for neutral lipids in the liver, and didn't really see any difference between the wild-type mice and the PNPLA3 wild-type transgenic mice. But when they introduced the I48M mutation into those mice, then all of a sudden you see this large accumulation of lipids. So there's much more staining in the panel on the right. And then they did a further study in vivo using a proteasome inhibitor and showed that the normal kind of wild-type PNPLA3 protein is degraded by the proteasome. If you treat animals with a proteasome inhibitor, levels of that protein will increase. However, if you look at the I148M mutant, the levels of that protein are already much higher than the wild type. And when you treat with the proteasome inhibitor, you see it somewhat of an increase, but it's already much, much, much higher than you see with the wild type protein. And so this led to the schema on the right here, where what's believed to be happening is that this large accumulation of PNPLA3 is actually competing for an endogenous lipase that's typically found on the surface of these lipid droplets and mediates the processing and degradation of lipids. And PNPLA3 is out competing it, so it cannot get onto the membrane of these lipid droplets in sufficient quantities to process all of the lipids. So you have a defect in lipolysis. So our goal then is, well, if we can identify patients, and this is actually a relatively common mutation in the grand scheme of things. So if we're able to actually identify patients that carry PNPLA3 that have NASH, then perhaps by reducing levels of the mutant protein, we'll be able to actually have a treatment for this subset of patients with NASH. So that's our goal, and this has been shown in theory by some investigators. I won't go through all of this data. Suffice to say that using a mechanism very similar to RNAi, they showed in mice that if you knock down PNPLA3 expression in a mouse that's overexpressing the I148M mutant, you can actually reverse the course of NASH in these animals. And that's what we're hoping to be able to do in humans. We've developed an siRNA to knock down PNPLA3 expression. And it does it in an allele insensitive way, so we're not specifically targeting I148M. Our theory is if this is simply due to too much PNPLA3, resistance to proteolytic degradation. We can kind of play the part of the proteasome, if you like, by reducing levels of the messenger RNA and trying to normalize levels of PNPLA3 within that hepatocyte. And so on the right panel here, you can see we screened various candidates. So what we'll do typically is we'll take the sequence of the mRNA of our given target, in this case PNPLA3, and we will tile short siRNA duplexes, which are typically 21 to 23 nucleotides across the mRNA, and we'll identify those that are most potent at knocking down the expression of the gene, and we were able to do that. We found several very promising candidates, and what you can see in the bottom panel here, again in non-human primates, is after a single subcutaneous injection, we're able to very robustly knock down expression of PNPLA3 in these animals. And so we have our clinical candidate. We're in the middle of doing GLP tox studies right now, and so we hope to go into the clinic with this later this year, actually. That's our goal. So, it's obviously, again, a challenging field, but this is one that we're particularly excited about because there's a genetic kind of definition of a population, which is something that's quite attractive rather than just, idiopathic NASH. It's an individual subset of patients that go on to develop NASH that carry the PNPLA3 gain of function mutation. So we are excited to test this in the clinic. And I will stop there and take any questions that you have.